Now we've got the reports session now, so we'll just go through what you're going to be getting in those emails that we send out to you, whether you're Marina Select or maternal, what you're getting back from us and how do we interpret that, and maybe something that you might be missing when you're looking at your reports. Okay, so yeah, like I said, looking at the reports you're getting and how to utilise those. And we do have a new report now available for you called the Reproduction Report. You can access this via the dashboard of when you're online on our sheep genetics page. You can go in and look at your flock information and find that reproduction report. Only if you're linked though, only if you're, like we still have thresholds on the amount of linkage or amount of information that you need to be recording to meet a reproduction report.
If you don't have enough information to go into the reproduction analysis to start off with,
we can't give you much back.
Yeah, we won't have a report.
Righto, so Marina Select, for those that are not in both, Marina Select get a bit different to what Land Plan happens to start off with, because when you send information in, they'll actually get a report coming back before the analysis gets run. Okay, so before it gets put in the run, we send back these reports. So the Marina Select members will get a database summary, a size summary, a pedigree, an exclusions, and a syndicates report coming back to you. Just to double check, like Karis was saying before, making sure we've got everything right, we're underway, we can put it into the analysis, and we're not going to look at it a week later when we get the reports back and wonder why these figures have changed and then found out we've entered a SI wrong. So just a little check-up before we put it in, and this doesn't happen for Land Plan. Okay. So when we send these reports out, you've either sent something into us, so sent in new information, and we've had to update your animals, and we'll send you back an email that will contain all these reports. Or the other option is when we do a report all. So we've done that recently with a few database issues in the Marina Select analysis. And yeah, so there's a few times that you'll get these opportunities and to see all these reports. And if you do want to update ones, just send us an email and we can add you to the list to get a report done on your flock, even if you haven't sent any information in. So keep that in mind if you do want to have a look at some up-to-date figures before joining, or a process that you might be looking at figures a lot for your sheep.
Is there any limit on how many reports you can actually request during the subscription?
No, you can get as many as you want.
So I could request it twice a month then, if I'm going to the extreme?
Well, we run the analysis twice every month. Those results are loaded to the website. So you can see all of your results on the animal search system on the website as well. We run 2 report hauls a year as standard, and then send out a report whenever you submit data. So if you do want others, that's fine, we can do it. The only reason might be that you haven't had a report for a while and you want the breeding value file to update your on-farm software, for example. So we can certainly send that out when you need it. If you haven't submitted data in between, there might have been data come in from related flocks, so figures might have changed to some degree. But you can, as I said, you can keep an eye on that stuff on the animal search on the website on the way through as well.
All right, so these reports that you get, you won't get them all, but most of you'll all get an individual listing and a size summary and a database summary report. So you'll get these 3 no matter what. And then depending on, and a genetic trend report. And then depending on if you had any issues, you might get an exclusion report as well, or a pedigree problem report. And if you're up to the stage of doing genomics, which is really, really good, you get an RBV report as well. So I'll run through those reports now, starting off with individual listing. You'll get a list of your size, your dams, and your male progeny and your female progeny for the current drop. Okay, so this is what we'll see. And this is a Marina Select example. So a few things you should be looking at is, 1, is the report current? Okay, probably not, this is a bit old. The next one, are my size linked? So see how we've got 171 here and then 1? That means he's got 171 progeny in one flock. Okay, so that's a Also another tip for if you're not sure if you've got the right sire typed in. If you've got him typed in right and you know he's been used across a whole heap of flocks, he should have a more than one here for the flock. If not, chances are you've made him up and we don't know that he's actually on another farm as well. So might be worth checking or keeping an eye out for anyway. Okay, next up we've got a little R and a little A here. So the first one is am I linked for all traits, and the second the A is for am I linked for all traits, and the R is am I linked for reproduction. So that's another little key step involved in working out if you're linked or not. So and that tells you if that sire is linked or if that progeny is linked. And again, another little key point is if this is blank over here, the sire, then yeah, you might have made up your own animal there that we don't know has been used elsewhere. So what to look for, you want correct dates on your run. You want to know your sire's been entered correctly and keeping an eye out for these A or the R, so if you're linked for all traits. So that's gold standard level, so top as best you can be, linked for all traits and you're linked for reproduction. That may not always happen. Okay, the second report that you'll see is a genomics report. So this little green star here that you can just see, that denotes that it's been genotyped, and you will also see some animals here showing in an RBV report that have not been genotyped, and that may be because they've been used or that their progeny has been sampled enough times that we're confident enough to give that sire a RBV. So research breeding values over here, we've got lean meat yield, IMF, so intramuscular fat, dressing percentage and shear force. Okay, so great. Inbreeding, I'll get to that in a moment actually. I'll get to that. So database summary, third report that you'll get shows what we can see in your database, and this is often a bit of a key report if you think you've sent in some information but we're not actually seeing it. So something might have happened between the space of you entering it into your computer and us receiving it and using it in the analysis. Okay, let's have a look at one. So first up, we've got birth type and rear type count. So you can see here, this property thinks that they've been having 92 born and 92 raised. Okay, so it is feasible, but if you know for a fact that you're seeing lambs dead in between birth and weaning, then chances are this won't equal that one. So if you're entering it properly, it should be a bit different. Or it could be right, you could be doing very well. Okay, so have we got parentage correct? Are we missing a few? You guys see the edge of that? It's meant to say 91, I think. Are we seeing a full pedigree list? So have we got dam, have we got sire? We've got all the dads entered correctly, but we're missing 2 mums that haven't been put into having a dam. 2 lambs are missing a dam. Okay, so like I was saying at the start of the day, you get half your genes from mum, half from dad. If we can only see how your dad performed, we're taking a bit of a stab in the dark for how the dam was. So, pretty key part. Okay, next point, are you recording a large enough proportion? So you can see here, we've started off with 123, and then we've only weighed, I think that state, or 115 we've only recorded at weaning age stage. So in between that stage and that stage, we're missing almost 10 animals. So it's in proportion, but if you get to a stage where you're not measuring, Hamish was talking about it before, if you're not measuring the bottom performers, that bottom 10, 20%, if you're not recording anything for them at all, we don't know just how good the rest of your group are, because you're not including those animals in the analysis. So we can't see that bottom end of the bell curve that we had up before. We can't see those poorer performers and where they sit.
And does it strike anyone as unusual that they recorded 115 ram lambs for weaning weight and 1 ewe lamb for weaning weight?
Okay, so that goes on to the next point of, have we got our sex entered properly? So according to this, we had a female with a scrotal circumference. So I think we've got something wrong there, and we do have adjustments for male and female. So if we've got a male entered as a female, chances are we're not interpreting it as best we can.
We don't have female adjustments for scrotal circumferences.
Yeah, we haven't got that far yet. Don't think we ever will either, will we? Okay, so that's your database summary. So look, a lot of numbers, a lot to take in on one page, I know, but it's, it can be a pretty key role in working out where you might be able to improve your data quality. Okay, so I think we've crossed all those bridges. Sex is right. Righto. Next report, so the size summary, and this brings up the inbreeding question from before. So we'll see how many size we've got. So first point off the rank was what's the inbreeding of your size? So this is just a bit of an overall snapshot of the size you've been using. What's their inbreeding at? Okay, so this is just for the size. This isn't for the year drop. This is just for the individual animal. So this fellow was 2% inbred.
Inbreeding, I guess, largely it depends on what your attitude to risk is. You know, there's a couple of impacts of inbreeding. One is inbreeding depression, so the opposite of hybrid vigour. So you're going to see that first and foremost in fertility traits, that sort of thing. The other concern is deleterious recessive genes, so lethal recessive genes, that sort of thing. You know, we know that they're out there. We know they're out there in all of the major breeds. Hypotrichosis is an issue in white Suffolks and Dermatosparaxis and so on in Dorpers. You know, I think the Angus breed's up to, 8 or so lethal recessive by now. The work that has been done looking at the 50k SNP chips that have been collected through the information nucleus flock, in each of the major breeds there, they've found between 2 and 5 genes that you only ever see an animal carrying a maximum of 1 copy of them. So suggests that they might be lethal recessives, that an embryo carrying 2 copies doesn't make it as far as birth. It's a lethal recessive. So we know that those things are there. The risk with inbreeding is that we inadvertently multiply up the number of copies of that gene in the population. So depends a bit on your attitude to risk and how you see those things. Depends where you sit within the industry. If you're right up the pointy end of things, you might need to be a bit more cautious about it because you can't go outside of your flock as easily to find other animals to solve inbreeding issues but keep your performance where you want it at the same time. If your animals are pretty average like mine, you can go anywhere and buy a ram that helps you out. So puts you in a little bit of a different position. But on an individual animal basis, if you see inbreeding coefficients up above 12% or something, you're going to ask why, what's happened there. On a flock level, if we start seeing inbreeding coefficients up above 5% on average across a flock, and that's trending upwards, then I'd be quite concerned about that. So, if you're seeing 1 or 2 animals at 2 and 3%, that's not a big concern. Keep in mind for merinos, we can only calculate inbreeding coefficients where we have got full pedigree in the database. So, just because you've got a 0 there doesn't mean you don't have an inbreeding issue. It might mean that we don't know enough about your animals to do it. Across the flock, it depends where you draw the boundaries. Kind of depends a bit on your attitude to risk. But personally, if it was above 5%, I'd be concerned and starting to ask questions about how we manage it.
And you also get inbreeding depression as well. The same way that heterosis and hybrid vigour works, it's the exact same if you're just breeding along the same way. You actually get a depression in the performance of your animals. So just keep in mind, keep it below 12. So we also want to know another little key point that you can take out of this summary sheet. Are your sires progeny even sexed? So according to this report, this guy here, he had 8 males and no females. So maybe true, maybe not. So It's a good gauge for you to go back and work out if you did enter those sexes correctly. And I think lastly, unknown size. So if we've got these ones down here, we don't know who their dads are, we're not being able to properly measure how their performance might be sitting versus where it actually is. So we really need to know as much about the pedigree as possible. Okay. So now the exclusions one. Has anyone got an exclusions list that they've received? A few people get them. So they're here to help you. They're here for a bit of an idea as to why we wouldn't be interpreting those animals properly. So, so the idea here, I'll just run from the top one. So if we've got outside of 4 standard deviations, we're going to be excluding just this trait for that animal. Okay, so a few people get caught up thinking that if that animal's been excluded, its whole performance is excluded. We're just on that trait. So, you know, someone might have carried out a typo or something. So if the average is 10, no matter what, or whatever trait we'd be looking at, say fleece weight, if the average was 5 and all of a sudden we see a fleece weight of 50 kilos, that's when we're going to say, well, someone's obviously had a typo there, we'll exclude it, let you have a look at it in your report, and you can go back to this later on.
So is the value for that animal correct? And is the management group that we've got that animal identified as running in correct? So in that case there, it's saying for fat depth, you've got a value of 5.5. The average of the group is 2.6 mils. The range from 1.5 up to that animal at 5.5. You know, that animal's significantly different to the rest of the group that it's being compared to. You know, has it been in the show team and had access to much better feed than those other animals, for example? We worked through with a flock just recently that had quite a lot of them coming up, and it turns out that lambs are actually born on a number of different properties and boxed together after weaning. So you've got all these different property effects that we're not accounting for in our group codes. So, you know, 4 standard deviations is very different to the rest of to the average of the mob. That slide Karis quickly put up before. 3 standard deviations, we would expect, you know, what was it, 0.1% of animals to fall outside of 3 standard deviations from the average of the group. So 4 standard deviations, not highly likely that that animal has a correct measurement and was actually run in those conditions. In the cattle analysis, in breed plan, you do actually have the option of going and verifying that that information is correct and having it put back in. Their guidelines of what's an outlier is a little bit different, but in this analysis we say, look, it's highly unlikely it's correct. Check the record, check the management group. If you can correct those, we can use it. Otherwise, we won't use it in the analysis. We'll use other records from that animal, so correlated traits will still apply and so on. In the cattle analysis, you know, I used to see people saying, yeah, that's right, verifying it. One herd that I did work for, large herd, you know, 800 odd bulls a year, they'd go through and verify those records, so they go into the analysis, get used in the analysis. That herd had the highest and the lowest 600-day weight breeding values in the breed because these outliers were being verified instead of actually going back and looking at the detail. Were these animals really run in the same management groups? As it turns out, after a fair bit of investigation, yeah, 800 odd bulls a year, they were run, you know, on oats with cell feeders in mobs of 50 or something, and they were saying, well, it's all pretty similar, so we'll call them all the one management group. In reality, you're seeing a fair bit of, you know, social hierarchies and that sort of thing that were influencing performance of animals, and we really should have been grouping animals differently.
Okay. So another exclusion, another reason for an exclusion is a date of birth not known. So we really need to hone in on those ones. So especially if there's only 2 there, we're a little bit worried about that. And again, like Hamish was saying, we've got this outside 4 standard deviations. We do have a weight to an eye muscle depth ratio that has a threshold. So if you've got a really light animal, a really light animal with a really high eye muscle depth, I'm not off the top of my head, sorry, I'm not sure what that ratio is, but we're looking at excluding that because it's not really that feasible to have an animal that small body weight wise, but to have an eye muscle depth that big. So again, this is coming back to these quality assurances that we're putting down to help with the information accuracy that we're accepting, okay? And like we've touched on already, if an animal's that far above the average, is it really being compared in the right group? Has it figured out how to get into the feeder unlike the others? So is there something happening there that you're not aware of? All right, so that was exclusions. And now pedigree problems. So this is from a Marina report. So we've got multi-birth dates. Karis was mentioning it before about having a ewe that has 12 lambs in a year. So if that's going to happen, we need to know it's from an ET program rather than just saying that she was naturally mated and somehow had 12 lambs in the year. And also people going back later. I think an issue does come up when people try to, people put in that there was a mothering up event a few weeks later. Depending on what software program you're using, I think there should be a field to enter in a foster dam. So a foster dam is separate to a dam, especially if there's a bit of a mix-up with who mum is. All right, what else have we got here? We've got no syndicate list. So here this person has sent in a named list, a named syndicate list, but we have no idea who was in it. So we need to be getting that information from you about who's in that named syndicate list for us to be interpreting it properly. And pedigree truncation, that's just where we're getting the year of birth being 4 zeros. So getting the year of birth right is enough reason to call for a pedigree exclusion. Yeah, we just got a question with Kate. We'll get this mic onto her.
I've got one animal that I put O for orange instead of 0 in it, and I've tried to change it, and I've emailed Stephen Field, and I seem to keep getting this report saying animal number A, B and C has got O for orange instead of 0. I've even deleted the animal and resubmitted the raw data, etc. I don't know what to do because... Okay.
Well, mate, we might bring that up later on.
Okay.
But yeah, make sure we'll have a chat about it.
I just keep getting that it's got O for orange.
Okay.
But yeah.
So yeah, like it is having in that having in a letter in the tag ID is a bit of a risky game, especially if you're going to maybe one day put in a CO or a CU. But we'll make sure we have a chat about that before you get home. Okay. So yeah, we need to know the syndicate list. Okay, another one that you get is your genetic trends. So this is going to tell you the run average and the breed average that you're being compared in and how you're performing against those, and the count of animals that we're using and showing if you're linked or not. So this is what we'll see. First up, linkage, we should know this. It's down here, bottom corner, whether you're linked. Yes, 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 all linked for those traits. So it is fair to be comparing. So using worm egg count, fecal egg count, we're linked for fecal egg count, so it's not actually on there. Reproduction, sorry. No. Okay, sorry, I was going down the wrong track there, but saying that was a...
Terminal cybrid, so it's only growth and carcass traits reporting on it.
Okay, so we've got carcass traits linked, yes. So we go up to eye muscle depth, and it is fair to be comparing where we're sitting against the breed that we are and the analysis. So this is a terminal breeder. The one line will be the whole analysis that you're in, and the other little grey line, some up the back there might just be able to see, is the breed. So 16 is Pole Dorset, and there's the terminal line, and then that's where he's sitting. But if this was a no, if this carcass was a no, it would be wrong to be relating where you're sitting against where we are for other breeds and for every other animal in the analysis, because you're not linked. So we don't know how you're performing for that particular trait. Okay. Sorry, I had a few other things to go through there. The count is down the bottom here. We've got how many animals you've sent in. So that can sometimes be a bit of an indicator as to why your trend might be going down, aiming into the ground, because we don't have all your animals in just yet. And that quite often is the case for the latest year. It's probably not quite as accurate as these ones back here, because we don't have as much information on them. So keep that in mind. We've got your averages per trait against the terminal. So if you're a white Suffolk, that'd be 23. So just a bit of an idea for that. Okay.